Assalamualaikum guys. Today our topic is characteristics of enzymes. Means what are properties of enzymes? Enzymes are protein in nature and are secreted by cells. What does this mean? Means that enzymes are made up of proteins and they are protein in nature and this enzymes is produced and prepared by the cells which are present in living body or in human body. That's why they are secreted by cells. Enzymes function as catalysts and increase the rate of chemical reaction. They do so by lowering the activation energy. What are catalysts? Catalysts are those things which increase a reaction. So enzymes are catalysts that increase a chemical reaction. An enzyme increase chemical reaction by lowering the activation energy of a reaction. Activation energy is an energy which is used during a chemical reaction to break the bonds between substrates or reactants. So enzyme lower of the activation energy and increase the rate of chemical reaction. That's why it is also called biological catalyst. Now, enzymes are specific in action. They are specific not only for substrate but also for the kind of reaction. What does this mean? Means enzymes are specific in action. They do specific action and they are not only specific for substrate. Means enzyme convert only a specific only one kind of substrate into product because it has a proper shape. We will learn this in the mechanism of enzyme action. There are two theories, lock and key model and induced fit model. So we will learn that how enzyme is specific for their action. So enzymes are specific not only for substrate but also for a kind of reaction. Enzyme only work in specific reaction in only one kind of reaction. For example, if we take example of pepsin, so pepsin only work in digestion. So this is his specific work. So, a small amount of an enzyme is needed to catalyze relatively large quantities of substrate. They are not used during chemical reaction. Means enzyme, if we take small amount of enzyme, it will bring change in a lot of substrate. Mean a small amount of substrate is useful for bringing change in large amount of substrate. And enzymes are not used in chemical reaction. As we learned in previous videos that enzymes kickstart chemical reaction without getting consumed. Means enzyme only kickstart or increase a chemical reaction but don't change itself in the reaction and don't take part in in reaction, and that's why this is not consumed in the reaction. All enzymes are synthesized inside the cells. Means if we as we in start of video we learned that they are secreted by cells, so it means that enzymes synthesize inside the cells. Enzymes are synthesized by our cells that are present in our body. Some enzymes work inside the cells and are called intracellular enzymes. Means some enzymes are synthesized in cells and they work and they work inside the cells. For example, mitochondrial enzyme. For example, mitochondrial enzyme, this enzyme is synthesized inside cell, but it work in mitochondria, and mitochondria is organelle of cell, means it is present inside the cell. So mitochondrial enzyme work inside the cell, and those enzymes which work inside cell, these enzymes are called intracellular enzymes. Some enzymes are released from the cell and they work outside. Means some enzymes are released outside cell, they synthesize in the cell and cell release their enzymes and they work outside the cell. So these enzymes are called extracellular enzymes. For example, pepsin enzyme working in stomach cavity. So pepsin enzyme, this enzyme is synthesized in cell but it is released outside the cell and this work in stomach cavity. So this is also a specificity of enzyme that pepsin only work in stomach cavity. That's why enzyme is specific for action. In complex metabolic reactions, many enzymes work together in a sequence. One enzyme takes the product of another enzyme as a substrate. After speeding up the reaction, the new product is passed on to another enzyme. means some reactions are very complex. So in these cases, complex in these cases of complex metabolic reactions, so many enzymes work in a coordinating system. They work in a sequence. One by one they are used in reaction. For example, one enzymes take changes in the substrate. After that, it is passed to the another enzymes, and another enzyme also work on substrate. And after that, it is passed to a third enzymes. So these enzymes work in sequence. For example, digestion is a complex metabolic reaction. There are many enzymes which is used in digestion. For example, pepsin, trypsin. So they are used in a sequence. Some enzymes require non-protein molecules called cofactors. Means some enzymes require non-protein molecule for their action, for their proper action, and these non-protein molecules are called cofactors for their action, for their proper action. Mean if they are not able to do action, so they attach some non-protein molecules with them, which is called cofactors, and after that they are able to do a proper action. The main types of cofactors are prosthetic group, coenzymes, and activators. There are three types of cofactors: prosthetic groups, coenzyme, and activators. Now we will see these three after one by one. What is prosthetic group? Prosthetic group are the organic molecules which make permanent attachment with the enzyme. Means this is organic molecule. Phosphatic group is an organic molecule and this is permanently attached with the enzyme. Mean it never detaches from the enzyme and it has a permanently attachment for the proper functioning of enzyme. Second one is coenzyme. Coenzyme are also organic molecules which are not permanently attached to the enzyme. Means coenzyme is also organic molecule like prosthetic group, but its differentiate between prosthetic group and coenzyme is that their coenzyme are not permanently attached to the enzyme. They have no permanently attachment with the enzyme, and after reaction they detach from the enzyme and leave it on life. And uh, when enzyme want it, so it attach with the enzyme during reaction. Most coenzymes are derived from vitamins. Means mostly coenzymes which are attached with enzyme, they are made of vitamins. This is also an MCQs. So this is a main point. Most enzymes are derived from vitamins. Activators. It is another type of cofactor. These are metal ions. Example zinc, copper or iron, which form temporary attachment with enzyme. It also form temporary attachment like not permanently attached, and this is another type of cofactor, but these are metal ions, for example zinc, copper or iron. Now there is a diagram in a book. This show that what is prosthetic group and coenzyme. Prosthetic group is attached permanently. For example, if the active site of enzyme is different from substrate, so prosthetic group is attached with the enzyme for their proper function to make is shape like same to substrate. So in this case, without prosthetic group reaction is not possible. And another diagram shows coenzyme. A coenzyme have temporary attachment, not permanently attached. So after reaction, it detaches from the enzyme. There is another thing in our book that enzyme inhibitors, like activators means that which that things which activates enzyme for working. So inhibitors is the antonym of activator. Enzyme inhibitor are those molecules that interact with enzyme to prevent it from working. As I say that it is antonym of uh, activators, means it is opposite of activators. Inhibitors prevent enzyme from working, like poisons and drugs. So these are example of enzyme inhibitors which stop enzyme from working, means prevent enzyme from working. So as I wish that you guys understand the topic, and please subscribe my channel and like and share my video.